So when we think about musculoskeletal function, we often think about whole body movement, right? And as you can see in humans and animals, there's a variety of movement patterns that are possible with varying degrees of agility. And we know that musculoskeletal function occurs via regulation from three primary systems. So our brain sends a signal down from the neural side to our muscles to contract, and then our bones react, right? And in some form, we generate movement. And if we think about what we do the most often every day, which is walking, there's actually quite a number of loading cycles that occur in the musculoskeletal system. And so in the United States, let's say we only get half of our steps in. So we have 5,000 steps per day. We have a life expectancy of about 80 years. And that comes to 392 odd loading cycles throughout our lifetime. And that's just general overall walking cycles. In the case of the hip, right, where your pelvis and your femur come together, there are 22 muscles that span the hip. And so that generates essentially 8.5 roughly million loading vectors that are occurring just on that joint. And so we're interested in understanding, how does the body handle this level of loading, right? And what are the determinants of making sure that our bones and our ligaments, for example, so we focus more on passive tissues in my lab, but hopefully by the end of this, I'll be able to convince you that the tissues are actually not so passive at all. But we want to understand what determines their quality. So with that in mind, we know that there are a couple of factors that we want to look at. And when we're talking about tissue quality, we're talking about mechanical tissue quality. So we need to know what the musculoskeletal loads that the tissues are expected to endure. We want to know how they're organized in terms of structure, and then we want to know what they're made of. There are many other factors, genetics, environmental factors, that we're not going to consider, but of course, those are also considerations. And because we don't want to do it the easy way, we want to understand something about the spatiotemporal complexity of it. And that's to say that these are not homogeneous materials. So they have some spatial variation in terms of the distribution of these properties. And we know that they change, right? As we go from little babies older into maturation, we know that the properties of these tissues are going to change. And so today I'm going to focus on two aspects of our work, looking at two different tissues. And in the first case, our goal is going to actually be to re-engineer bone. And so I'll just leave that as the teaser for that. And in the last case study that I'll talk about towards the end of the talk today will actually be about reverse engineering ligaments. And this is sort of a reverse bio-inspired design project. So let's first start with bone. So form and function in bone has a long history. And Carl Coleman and Georg von Meyer were at a conference together and realized that the principal stresses that Coleman, a structural engineer, saw in his femurs, in his cranes, seemed quite similar to some of the same principal stress directions that we observe in the proximal femur, again, this bone at the top of your hip. And so if we just apply the loads that are shown in the red arrows, we can see that we have principal stress lines, right, that have very specific orientations, but we know that we can see this architecture, right, in the trabecular bone here, which is that spongy inner bone on the inside. And so they correctly noted that the arrangement of this cross-section is most likely due or influenced by the muscle and ligament, essentially the musculoskeletal loads that are applied. And so a graduate student in my lab, Michael Rogalski, wanted to really understand whether or not this was true, and actually questioned some of the fundamentals of this assumption. And specifically in this picture here, these red arrows are really more showing the joint contact loads. So we have muscles attached to our bones, and those are countered by hip or by contact forces between the two bone segments. And in this case, we're only looking at half of the picture. And so the question was, what's the contribution of the muscles? So we went back to an older data set that I have where we evaluated muscle bone function using coupled musculoskeletal and finite element models. So we collected gait data on postmenopausal women. We have musculoskeletal models that can give us predictions of the muscle forces. And then we can combine those together with finite element models based on medical images of their femurs, and then look at actually what the contribution is of these different loading conditions are in terms of the strain in the bone. And so Mike did a very simple but important computational experiment of looking at the difference. What is the difference in the strain and principal strain distributions and directions when we have a model that only has just the hip forces, now shown in purple, and when we include the actual muscle forces in the model? And we only looked at one cross-section in the femoral neck. Somebody said I can try this other laser pointer. Okay, great. Hopefully that works. And so we looked at just the cross-section here within the femoral neck and split it up into 4 different quadrants to see what the strains actually were, looking at their magnitudes and their distributions. And so showing just a few of the important results, again, the purple was the hip-only model and the orange is the hip and muscle model. We see that there are decreases in the posterior aspect of the femoral neck in terms of the magnitude of the principal strains. And in the lateral quadrant, which is that superior side where we see some of the more preferentially oriented trabeculae, we see pretty substantial differences in terms of the magnitudes of the principal strains. So on the order of about a 50% increase or decrease in terms of max and min values. Of course, what we were sort of a little bit more interested in is about the orientation and whether or not the orientation of the principal strains changed, because this would then, in theory, have an effect on what we might expect in terms of the trabecular orientation. And what we can see is that when we include the hip and muscle forces, we see a significant shift in the direction of the strain. So each of these purple arrows represents the the orientation of the principal strain vector, the maximum. And we can see that if we only have the hip forces, they're basically pointing up, so proximal, but when we include hip and muscle forces, then they actually move more medially. In terms of the lateral aspect, again, there's a slight shift, right, between the purple vectors and the orange vectors, showing that there is a difference in the orientation. Now, all of this is to say that if we actually look at what's happening, these muscle forces are decreasing the bending moment. And if you draw the free body diagram in your mind, you might be able to see how this is true. If we have a fixed end here in the hip model and we only apply the joint load, we're definitely going to get a compressive side and a tensile side within the femoral neck. But if we include those muscle forces, we definitely see a difference. And all of this is to say that muscle forces really do matter. So why do we care about this, right? And how do we use this information? What we're interested in terms of quality is treating diseases like osteoporosis and understanding why bone becomes fragile or is more susceptible to fracture as people get older. And these are our updated numbers, and we can see that the mortality rate still for those who sustain a hip fracture, so this is evident in the x-ray on the right, still have about a 25% mortality rate within a year. And there are still long-term effects as a result of a hip fracture. One of the reasons we think this is because we know that the detection of poor bone quality, so if it's mechanically compromised, often comes late in life. But we know, right, that bone is responsive to loading. And some of the strongest evidence that we have for this is some of our work in baseball pitchers. So we followed, if we look at a baseball pitcher, an active baseball pitcher, we can look, for example, at the difference between their throwing and their non-throwing arm. And the non-throwing arm is shown on the left, and the throwing arm is shown on the right. These are 3D reconstructions of CT scans. And if we look at their cross sections, we can see very clear differences between the non-throwing arm and the throwing arm. Now, mind you, this is an active baseball pitcher. And the question is, what happens after they stop pitching? So if we look at the non-throwing arm now shown on the top and the throwing arm, over time, the throwing arm will eventually start to look more and more like the non-throwing arm. But if we look closely at someone who hasn't pitched in 50 years, we can see that there are still some structural adaptations that are maintained. And that is that the overall dimension of the bone in the throwing arm compared to the non-throwing arm is still larger. And this means this bone is still stronger. What this tells us is that some bone adaptation that can happen when you're young has the potential to last lifelong. So even though they haven't pitched in 50 years, we still can maintain some of the effects of exercise when done young. Now, the caveat to that is that baseball pitchers, of course, these are professional baseball pitchers. This is an extremely one-sided sport. So the level of adaptation that we see here is due to an incredible amount of load on the humerus, so the bone of the upper arm. And they have likely been pitching since they were very small, right, in order to get to professional levels. And this probably is not realistic for the rest of the population. And the question still remains, how do we translate this to benefits at areas that are prone to fracture, like the femoral neck that I showed you in the previous slide? So we looked in the literature to understand, how does the femoral neck grow during growth, and then what's the effect of exercise? And there are studies that have used aerial bone mineral density, so not as refined as CT, but that give us an indication of what is the femoral neck bone mineral density during growth. And so this was some work recently done by Chinchi Yan and Sarah Masaga in our group. And here we're plotting femoral neck aerial bone mineral density on the y-axis, and on the x-axis is age. And what we can see in the males in orange and the females in blue is that we have a steady increase in bone mineral density that plateaus a little bit earlier for the females compared to the males, and then slowly starts to recede. And we looked at what's been documented in the literature in terms of exercise and adaptation for sports. And we can plot and characterize exercise in two broad ways. We can think about the static component of an exercise, and these are basically activities where you can develop a large amount of force, but very small changes in muscle length. And so rock climbing is a good example of that, where you're holding on for dear life if you're going free solo style, but you're not really necessarily inducing a lot of motion, right? And then dynamic components of exercise are those that actually develop small amount of force, but repetitively and over a longer sustained duration of time. And so we looked in the literature to see what was out there in terms of how does exercise affect femoral neck bone mineral density, for example. And these are some of the activities that we found documented. So I'll give you a second to look and find your favorite sport and see where you fall on this diagram. Obviously, there are some sports that have both a high static component and a high dynamic component, such as cycling. And now we can plot these on this control. So on the current plot on the right are our control baseline values. And we can see that there's a bit of a variety in terms of what's possible for adaptation. So for example, it seems like the dynamic sports like soccer might actually induce an adaptive response. So this little orange dot is significantly higher than our baseline controls. But we also see that static values can also be higher, and that's the case with these gymnasts. What's been a bit more difficult to assess is whether or not we see that similarly aerobics, in this case a high dynamic sport, actually has decreased properties. So this little blue dot right here relative to the baseline value is actually lower. And so this begs the question of what is really the relationship between muscle forces, exercise, and bone adaptation, and do we really have a good handle on how those three things work together? So our premise, given that we know adaptation can happen, especially when it's done when young, is to understand what can we learn during growth that might actually have some benefits for us later on. Now, this is difficult to do in humans, but we can start by looking at equine athletes in this case. And this is, again, a collaboration with Dr. Annette McCoy, who's a veterinary orthopedic surgeon here in VetMed, and Sarah Masaga, an equine enthusiast and a fantastic grad student in the lab. And when we look at equine athletes, we know that over half of the racehorse fatalities that occur during racehorsing result from a fracture, and that those stress fractures occur in training, and that training begins around 1.5 to 2 years of life. We know that there are lesions documented in the literature that tell us that these fractures do seem to be fatigue related, and that they tend to happen here in this metacarpophalangeal joint, a bone down here at the bottom. a joint at the bottom that's made up of the MC3, the third metacarpal, and then the P1. And we're going to focus on the proximal phalanx today. So our first question is, what's going on with these horses and why are they susceptible to fracture? When we look at what foals do, we might think that, they're walking around in their pastures, gloriously gallivanting around. Here's some video that we took of a foal when we did some motion capture of them following the mother. But in fact, the reality is that they tend to be a bit lazy in their first year of life. So using just observation and cameras put out in the pasture, we found that foals spent the vast majority of their time eating, nursing. So this is, of course, a quiet standing activity, just standing or laying around. And about less than 1% of their time is actually spent at any sort of speed levels in terms of trotting or running, and about 10% of their time is spent walking. And we can see here from this picture alone, exhibits A through E, in which we have horses or foals that are just laying around. So what Sarah did was tracked a small cohort of foals as they grew from 7 weeks of age to 57 weeks of age, and looked at what happens in their bones and to understand how does it adapt in general to the process of growth. And then from there, the question is, can we modulate this? So we did longitudinal CT scans, included mineral density phantoms so that we could look at the distribution of mineralization in the bone, and then split them up into these functional quadrants to analyze how their structure was changing over time. We know that about 60% of their body weight is on the front half of the horse, and so given the fact that they're just standing there, we can assume that their basically functional role at this point is just to resist gravitational loads. And so we developed finite element models orienting loading conditions, since the muscle activation would be quite low during quiet stance, just applying the joint reaction forces at the angle that's been observed in X-rays. And then looked at the difference between the cortical bone on the outside and the trabecular bone on the inside. When we looked at the finite element models to see what the difference is in strain energy over time, we found a couple of interesting results. On the x-axis here is mass. So this is, of course, as they're growing from the smallest foals to the oldest foals, essentially on the x-axis, and strain energy density in 3 different regions is plotted on the y-axis. So we looked at this distal region, the diaphysis, and also in the proximal region where those fractures tend to occur. And we can see that the strain energy density is highest in the proximal region for both cortical and trabecular bone, also high in the distal region, but quite constant and steady over time in the diaphysis. So we wanted to look at what adaptations are occurring in the diaphysis versus the proximal region that would explain these differences. If we look first at mineralization, what I'm going to plot here is on a polar plot the rate of mineralization or the change in mineralization in the 4 different quadrants that we talked about before. So on the inside, the lighter colors represent the younger foals or lighter foals in terms of mass, and then the darker colors represent the heavier foals. So obviously they're likely to be older. And what we can see when we look at mineralization, it starts fairly uniform in all 4 quadrants, so it's almost circular. And as the foals grow or increase in mass, it grows equally uniformly over time. And so if we plot the mineralization rates as a function with respect to mass, we can see that they're fairly constant in all 4 quadrants. Now, if we look at the proximal region, we see a small variation in that, right? And that's that the mineralization levels in the dorsal and medial quadrants, up here are the sort of north and east quadrants, are higher than in the palmar and lateral quadrants, right? If we look at trabecular adaptation or trabecular mineralization rates, we see the opposite. So we see that trabecular bone starts highly mineralized when young and then becomes slightly less mineralized during growth. And then again, in the proximal region, though, we do see the opposite trend where the trabecular bone is gradually mineralizing. Likely, it's possible converting to cortical bone or losing some of the trabecular bone. We can also look at differences in bone fraction. So how much bone is there? And what we see is that bone fraction changes are drastically different in the diaphysis versus the proximal aspect. So here we're starting with, again, the less mass / younger foals, and we see that roughly, again, sort of a constant level of accrual of bone mass, so the amount of bone or bone fraction that's there. And then if we look now in the proximal region, we see a very different story. And now here we can see this very asymmetric pattern. So the initial state is this small light blue circle here. And then we see, pretty drastic increases in the amount of bone present in the dorsal and medial regions, but almost nothing within these lateral and palmar regions. In terms of the trabecular bone, we see something equally interesting. So this is a little bit more asymmetric. Again, the trabecular bone is decreasing, so we think the trabecula is eventually converting into cortical bone. But when we look in that proximal region, we again see what initially starts as a quite symmetric pattern. We essentially have all trabecular bone being formed right within these lateral and palmar regions, and a decrease in trabeculae in the dorsal and medial region. So there seems to be some preference in terms of when we're going to have cortical bone and trabecular bone in terms of the locations. Now, what's interesting to us is that it's this very region here where there's no cortical bone or very little cortical bone relative to the other quadrants is where these fractures occur. But of course, this is normal growth, right? So why would this bone actually grow in this asymmetric way? And we don't have a good answer, at least scientifically, but we have some guesses, and that's that the horses, as they're feeding and eating, they're not necessarily right under straight compression. And so there is offloading of the bones, and maybe there's some interaction here between the posture of the horses that they adapt as they grow, that they use as they grow, and the actual adaptation of the bone. So in terms of form and function, what we see is that we have heterogeneous distributions of mineralization and the amount of bone, and that functionally these may be correlated with some whole body biomechanics postural differences. The next question is what happens during locomotion. And so here we have baseline. Now that we have a baseline longitudinal characterization, we wanted to know what happens when we actually run these horses. And I'm just showing you, these are sort of very preliminary results, but they're really inspiring for us because they do tell us a little bit about what we might be able to predict in terms of the bone response to exercise. So we ran, again, another set of models where we now imparted the loads from walking and trotting, just ground reaction force-based loads, essentially propagated up through the P1. And what we see is that this lateral side, where there's little cortical bone, actually does increase in strain energy density. And so this would tell us if strain energy density is a possible mechanism, right? It's thought to be a mechanical stimulus for bone adaptation. Then if we walk them or run them, we might be able to see some of that adaptation. If we look, so now what I'm plotting here is our baseline data in the solid line with the standard deviation bars, and the actual data points here are horses that were exercised or foals that were exercised. So the pre-exercise values are with the open symbols and the post-exercise are with the solid symbols. And what we can see is that we have some data points that are not falling within the standard deviation of our control data in terms of bone area fraction. This is in the diaphysis in the proximal region, looking specifically at that north dorsal quadrant. And then we also have some diaphyseal adaptations in the lateral quadrant and not quite as much in the proximal region. So they are somewhat off the control lines, but it's still not 100% clear if we've done anything in terms of the proximal adaptation. We have very similar results in terms of density. And so this makes us question whether or not spatially targeting exercise protocols are really possible, because our goal, right, is to use less of a trial and error approach in terms of helping bone adapt and doing something more mechanically informed that would tell us which of these exercises, turning, running this way or that way, walking, trotting, and at what speeds, what strain rates would actually induce an adaptive response. So it's clear that not necessarily all exercise is going to induce adaptation where we want it. And this has also been seen in humans. So there have been some exercises that induce no femoral neck adaptation, for example, and then there are some exercises, and as we saw in the previous slide, that do. And so we wanted to understand whether this is really mechanistically possible. It's not easy to do this, again, in humans or in horses, but we ran a large animal sheep study to evaluate the effect of exercise during growth, and specifically two targeted interventions. So in this case, this is work done by Dr. Kyung-Mi Song, a recent PhD graduate from our lab. And in this study, we exercised 30 healthy juvenile sheep. There were non-exercise controls. There were some sheep exercised on a flat treadmill, and then there were some that were exercised on an incline, and the exercise period was for 60 days. During the course of the study, we recorded motion capture and ground reaction force data. So the motivation for whether or not we run them on an incline or not has to do with actually trying to target the actual mechanical loads. So in flat exercise, we know that we expect a broader range of loading of the joint compared to the incline exercise. And so we expected a smaller range of motion because when you're up on an incline, you have to turn your legs over a little bit faster, and the actual points of application of load we expected to be smaller. And so we thought the adaptation would actually be smaller. At endpoint, we imaged the distal femur. So this is the end bone of the femur here. This is the tibia, and took micro CT scans at 50 microns from these bone segments. All of the segments were aligned using anatomical landmarks, and then included, again, hydroxyapatite phantoms to calculate apparent density throughout the bone. And then the images were binarized and separated in terms of subchondral thickness and trabecular bone. So what we did here is we actually extracted different layers of the bone. So similar to sort of think of peeling layers of an onion, we analyzed first the subchondral layer. So that's this sort of dark, brighter white area, and separated that from 3 different layers of the trabecular bone, moving in towards the center of the joint. With these layers, then, we could reconstruct, and we did this over the entire femoral condyle, we could reconstruct 3-dimensional maps that let us evaluate each of these different layers of bone properties to see what the effect of exercise was. Now, it's a lot of data, so I'm just going to show you some of the big picture overview of this work. And what we're showing here are the average maps across all of the different subjects. So we have a control group. So this is the average of, for example, thickness at 4 different layers, from the subchondral bone shown at the bottom, and then deeper into the condyle shown at the top. Red is just more of the parameter and less, and blue is less of the parameter. And here is just a map to orient you in case you're curious about the orientation of the map. And what we can see is that in general, bone thickness was thickest in the subchondral region and then decreased as we moved deeper into the joint. And we can see that there were differences in the flat and the incline group, and I'll show some of the quantitative data on the next slide. But this is just so we can appreciate the variation in properties that we see. Similarly, we have variation in density, so it's not necessarily uniform, but it is decreasing as we move towards the center of the joint. Bone volume fraction, not surprisingly, was higher in the trabeculae near the subchondral bone and lower in the deep zone, so this is likely due to that increase in thickness that we see. And then degree of anisotropy, so the orientation of the trabeculae also had a heterogeneous distribution. So what we really want to know is whether or not what the effect of the exercise was, right? And what we saw is that there were actually more changes in one aspect of the bone than the other. So what we're showing here is over a single region, moving from the anterior side to the posterior side, so throughout the flexion arc. 2 different properties, thickness and density. And we have in 2 different regions, subchondral bone, and then the thicker, the most inner trabecular zone, what we call zone 3. Now, the absolute values of the thickness and standard deviations are shown here in the plots. And then the percent differences that were significantly different along the joint surface are shown in these dot plots here. The take-home message here is that exercise increased subchondral thickness by roughly 25% for both flat and incline, and density increased by 12% in the flat group, but not in the incline group. In terms of the trabecular zone, this is where we saw the greatest changes. We saw increases in bone volume fraction and thickness within this middle zone, but not in the zones towards the subchondral surface. So let me give you an overview of what these results really mean in terms of the different layers. So here we're showing our example of a control, just general cross-section. And so we have the subchondral bone shown in red, the next layer of zone in orange, green, and blue, and so on. And essentially the effect of exercise was that we saw increases in both thickness and density in the subchondral bone closest to where the load was applied. We saw next increases in density, and then this green zone in the middle, for some reason, has no difference that we can see. But if we move yet again a little bit closer into the joint surface or into the condyles, the center of the condyles away from the load, we see that there were increases in structure. And so there's this interesting binary response where sometimes density is increasing but not structure, and sometimes structure is increasing but not density. And this sort of reminds us of the horses again. And so the last thing we wanted to do was look at the gait and understand whether or not there were any associations in terms of where the loading was applied. We know that the range of motion in the incline group was in fact lower than the flat group, as we hypothesized would be the case when you're on an incline treadmill. And we can see here, for example, the thickness map of the flat group. This is again in the subchondral bone compared to the incline group. And we see that the region of increased thickness is actually smaller in the incline group compared to the flat group. And again, this trend exists in terms of density. One thing that we might be thinking happening that's a little bit been counterintuitive is why the density in the incline group, which has higher ground reaction forces and more concentrated loading, is actually lower. And it might be because the bone is still undergoing mineralization. So if it's induced a response where the bone is now trying to adapt, it might be that we are still in the middle of a full remodeling cycle, in which case adaptation would occur by an initial laying down of undermineralized bone that would eventually mineralize over time. We're looking at some of fluorochrome data to actually see what some of the modeling and remodeling rates are, but we don't have any of those results at this time. The last part of Hume-Lee's work was looking at whether or not any of these changes were mechanically significant. And so here we took a subset of the samples, 5 from each group, and then essentially cored out the regions that we saw the highest amounts of change within the bone samples. And so now we're just looking at a small cord region of bone. The samples were preconditioned and at 1 Hertz, and mechanical testing was performed. So here's a general overview of what our stress strain curves look like for these compression tests. And you can see the control group is shown here in blue, followed by the flat group here in this reddish color, and then the incline group is in yellow. And I'll talk about the individual parameters here on the next slide. Essentially, the effect of exercise increased the yield stress and the elastic modulus more so in the flat exercise versus the incline group. And we see that the resilience and the ultimate stress was increased only in the flat group compared to the controls, but not in the incline group. When we look at strains, we saw no differences in the yield strains, no differences in toughness, and a decrease in the ultimate strain in the incline group. And an increase, in toughness to 7.5%. Oh, okay. But I think one of the take-homes that we're really excited about is that these small changes in structure and density that we observe, so here an increase in subchondral density of 14% and a nominal increase of 11% in trabecular thickness, resulted in these really significant changes in terms of the mechanical properties. So increases on the order of greater than 70%, as you can see here, for both the yield stress and the elastic modulus. And the reason we're excited about this is because if we think back to the horses and whether or not our exercise data has an impact, whether or not these small changes that we see, while these data points are just barely off of our control line, these might actually have some meaningful effects in terms of mechanical properties. So the next question is what happens with the cortical bone? And this is some work done by Soni Manandar in our group. And she's been looking at what's happened in the same group of sheep in terms of the cortical adaptation. And she's created density maps and cortical thickness maps, again, and along the whole bone cross section. And now we've decided to look at 10 different regions throughout the diaphysis to look at where there might be an effective exercise. What we see is that there's a effective exercise in terms of cortical density and in terms of increased cortical thickness. But again, this is spatially heterogeneous. So density increase in the posterior quadrant, whereas thickness and bone area fraction increased in 2 different quadrants. And again, it's this interesting dichotomy of why bone is responding in these 2 different ways. We also see a significant increase in the porosity of some of the bones. And so we're interested in looking at whether or not there's increased vascularization as these bones are subjected to exercise loading and what that might mean. In terms of the relation to functional loads, we've taken an initial stab at looking at what the strains might be in vivo. And of course, we do this using computational methods. So we take a CT scan, couple it with a musculoskeletal model. And in this case, we just took some data from the literature about what the forces are at the 2 ends of the bones and a couple of simplified muscle forces. And now we can look at what the relationship is between the adaptation and actual strain values. And what we see is that in this case, the femur during walking at the time of the peak stance phase is actually in a state of bending, right? So we have tension on one side and compression on the other. And if we look at the sheep, we can see that this is maybe not so unrealistic given that bone's orientation with respect to the loads that it might see and the fact that these are quadrupeds. And so now if we look again at our cross sections and look at the difference between the anterior side, which is in tension, and the posterior side is in compression, we can see whether or not there's any relationship here. And here we found some interesting things as well. So what I'm going to do is plot the percent change in terms of first bone volume fraction in between the flattened incline group versus the controls. And I'll plot increasing tension to the right. So we're essentially analyzing the samples moving proximal to distal, knowing that they're experiencing more tension in that direction on the anterior side, and then increasing compression on the other side. And what we can see is that the largest response in terms of bone volume fraction actually happened in the bone subjected to tensile loads compared to the compressive loads. And this was a little surprising because bone is known to be stronger in compression versus tension, but perhaps this change in loading state might actually be inducing more of a response than we had originally anticipated. In terms of density, if we do the same thing and plot the adaptation on tension versus compression, we actually see a slightly different response. And that is that in the same areas of increasing tension that induced a significant increase in bone volume fraction actually induced, again, no change or even a decrease in density. Whereas increasing compression and low levels of tension all seem to induce a density response that was fairly uniform. Again, we still need to investigate whether or not the fluorochrome labels that were induced would actually explain whether or not we are in the middle of a remodeling cycle, or whether this is really, in fact, a true response in terms of decreased density. All right, so that's what we've learned in terms of our understanding of how to re-engineer bone in terms of its functional adaptation to physiological loads, in an effort to prime it for whatever function it might need to sustain. In the case of the horses, for racing, and hopefully for people at some point we can translate these results to developing better exercise interventions that we might use when young and during growth, during maturation. So I'm going to switch gears in the last couple of minutes that I have and talk about our reverse engineering project, which is related to ligaments. And so ligaments, again, are a passive tissue in the sense that we are responding to loads, but in obviously a very still complicated multi-scale structure. And this project was motivated by actually a collaboration with Jump Arches over in Peoria, the Simulation Center, in which they wanted us to start to look at whether or not we could develop a knee simulator. And the fact is, knee injuries are everywhere. And so here's my pity Twitter post where I think it's on July 22nd, I was jumping on a trampoline with my daughter, and my knee just kind of buckled out from under me. And it turns out I, in fact, now have an ACL tear. And knee pain is among the most frequently reported joint pains. And the current standard for detecting a knee injury is difficult to assess, and it's difficult to teach and to learn. And so while our primary care doctors are usually our first point of call before we get a referral to orthopedics, they are still a little bit hampered by the inability to necessarily perform the procedures properly, and hence the referral to orthopedics to confirm a diagnosis. And some of the scary numbers here are that half of the family physicians, this is from a study in 1977, thought the training in orthopedics was inadequate. More recently, there's been demonstration that third-year medical students, and then by the time you get to internship, 61% fail to demonstrate basic understanding of musculoskeletal problems. And while this number is a little bit old, half of American medical schools did not require a musculoskeletal course as of 2003. And so when we think about how do we better train our doctors, we can look at what are they using right now. Obviously, they're using each other, they're using standardized patients, they're using whatever they see in the clinic, and then perhaps more commonly are some of these just aesthetic models of knee function. So here we're looking at what you might see in a doctor's office. And so you can get these for as little as $28 here, a knee joint model to demonstrate the contributions of all the different structures of the knee. So this is mostly ligaments, muscles, and bone in this case. And you can fork out an extra $100 and get a deluxe version, which to me looks a lot like the $28 version. But you can see that these are aesthetic models for viewing purposes, but not necessarily functional. And so the goal here is to actually develop a biomechanically realistic physical knee model that could simulate the learning experience. And in order to do that, we need to understand how do we create an artificial ligament then, and not in the context of necessarily cellular engineering context, because obviously these would just be teaching tools, right? This is very different, right, from a cell engineering approach. And so our goal was to understand whether or not we could develop a synthetic ligament that would replicate the mechanics of knee ligaments in order to build this model. And so here's my pro tip. This is a slide from my PhD and I encourage everyone to save their data and save their slides, because we were able to tap into, again, an older data set from my PhD where we evaluated knee properties and ligament properties at different resection states. So we have an intact cadaver, for example, we can measure their mechanical properties, remove skin and muscle, and then sequentially understand the contribution of each of the different structures within the knee to evaluate not only their mechanical properties individually, but also how they aggregate together to provide stability and function for the knee joint. And so this is where a student in our lab, Roberto Pineda, has been working to understand the complex multi-scale behavior of ligaments, right? And so we've all seen, or many of us have seen, this kind of diagram to look at the different multi-scale structures that exist within a ligament or a tendon that's often also used. And what we know when we look at the stress-strain curve of these tissues is that they exhibit nonlinear behavior, so kind of characterized as a J-shaped curve. There's hysteresis shown here. And if we look at just a single collagen fibril, or a set of collagen fibrils, we can see that we get nonlinear behavior at every single length scale. And if we move up to the fiber level, we still see this exact same behavior. And so our question is, okay, how do we use this information to architect this artificial ligament, and what should we make it out of? So there are some really great labs on this campus looking at bio-inspired design. So this is just two examples. There's the BAM lab, one of the best lab names ever, from Professor Amy Wissa in mechanical engineering. in which they use bio-inspired design for different robotic applications. Professor Rainer Johnson has also developed artificial scaffolds, for example, for treating bone defects that have some inspiration from the natural porosity of bone. But what we're looking for is more of a reverse bioinspiration, right? I want to find a material that just so happens to have similar maybe structures or mechanical properties as this real material. And so we were sort of inspired by looking at the kibben muscles, and a fantastic undergraduate, Sam Goldsmith, helped us kickstart this project by essentially scouring the literature looking for artificial materials that had that J-shaped curve. And Roberto picked this project up and really started to run with it. And what we found is a really simple solution. And so this is why I'm so excited to talk about this here in my last few minutes, because it's been a surprisingly complex and simple solution all at the same time. And the material that we've honed in on is yarn. And the reason for this is that yarn displays some of these very same complex hierarchical structures. They're woven materials, they have a specific organization, they're wrapped in certain ways. And what we wanted to then understand is at what length scale these materials would exhibit the behavior that we want. And so we could look at a 4-ply yarn and pull on it in the Instron machine, and we immediately saw that we got this nonlinear stress strain curve. And we were really lucky because our first yarn that we tried was actually ended up being the most successful, spoiler alert. If we peel apart the yarn and look at it at different levels, we still see that there is some of that nonlinearity, but there's something magical, right, with this. There's not much difference in terms of the 2, 3, and 4, but if you have a single ply of yarn, you get this increase in the parameters. And so we wanted to know what's causing this J-shaped curve specifically in yarn. And Roberto was very careful in his experiments and just observed what was happening as the yarn was being pulled. And it turns out that as you start to pull on the yarn, you initially get this low toe region of the curve here. And as we keep increasing, what we get is essentially contraction of the yarn. And so as the force is starting to increase, the yarn sort of contracts, right? Sort of as we would imagine like a Poisson ratio, but it's not because of a solid material, it's because of the arrangement of the fibers. And so like many great studies, we sort of detoured over into the world of fiber mechanics and looked in the textile industry. And there is a bulk of work out there that's looking at how does yarn actually develop its mechanical properties. Interestingly, in the literature, they usually don't care about that low strain, this region here, P1, but for us, this is where a ligament is the most functional. By the time you're out here, you're kind of at the ends of where your ligament tension would be expected if this is our ligament surrogate. But we know that that compressive pressure in the yarn is related to a couple of things that we could actually measure: the fiber modulus, or look in the literature for packing fraction, so how much yarn there is within a given area, and then, like any good model, a proportionality constant to help us. This data can be used to relate the yarn stress and the yarn strain. And the only point really for this slide is that we can actually measure some of these parameters. So we can get an estimate of the fiber modulus and look at the fiber twist angle, theta, so how twisted these are, and then again, how many yarn fibers are packed within the cross-sectional area. And we looked at 4 different yarns. So here's just, again, an example of the stress-strain curve where we looked at both the toe region, the initial low stiffness region, and then this high stiffness region called the heel region. And then we started imaging our yarn. So we measured the fiber twist angle, the apparent diameter, and the ply twist angle. So there's, twisting upon twisting in this case. We then micro-CT'd the yarns by embedding them in silicon and then analyzing the data to actually get some of the packing fraction values. And so these are some of our results, and we can see here each color represents different yarn types, and we get very different mechanical behaviors depending upon the yarns that we're looking at. And what we can see is that essentially the packing fraction, how much yarn in there, is really the primary determinant, right, for why we would get a low stress tensile behavior within the yarn. that packing fraction is also related to the twist angle. And again, our one yarn that exhibited the best properties stood out in terms of having the highest packing fraction and the lowest twist angle. And so together, what we've been able to do now is create different ligaments. And so here again are those 4 different ply levels. And I'll just call your attention to the figure on the bottom right, where here's our cadaver MCL back from my PhD a long time ago now. And now we've been able to know how exactly to fit our yarn parameters and the length of the yarn, the number of loops that we would use to best match this specific behavior. And together, we've put this together in what is our first generation prototype of this of these knee models that will be used for simulators for medical education. Roberta is also working on some really great work looking at in vivo imaging of yarn, in vivo imaging of ligaments to actually apply some of the fundamentals in terms of packing fraction that he's observed in the yarn, and whether or not, that provides a good foundation for us to look at some of the microstructure within ligaments. And so most recently at Beckman, we've been able to successfully, using diffusion tensor imaging, getting some mesoscale microstructural information in ligaments. And so here you can see these fiber tracks, similar to sort of our yarn tracks that we've seen before. So hopefully, I've shown you some ways that we're interested in probing some of the determinants of tissue function, especially in terms of their structure and composition. And we, are going through these cycles of looking at a combination of experimental and computational techniques to answer what we think are some important questions, in particular clinically. Again, I must thank the students who have done all of the work that I've shown here today. I'm sorry Hyung-Wee's picture is not on this slide, but obviously some of the sheep work was done by him and our funding sources. I'd be very happy to answer any questions that folks may have.
Hi, Mariana, this is Fan. So
Hi.
Great talk. So I just have a quick question from me. How did you get the sheep to exercise willingly on the treadmill?
Yeah. That's a good question. Essentially, animal science students have to be trained in animal husbandry of handling animals down at on the sheep farms, and so they're acclimated to the treadmill, and get used to it. We start them a couple weeks before we intend to start the exercise program, and they are quite skittish. But once they get used to it and know they're going to get a treat at the end, they comply.
I see. Cool. And I also noticed a question from the audience in the chat. Someone asked during your talk about whether you have considered using stem cells to study the
ligaments,
bone
maybe.
function. Yeah. Yep.
Yeah. So that's an interesting question. For this application, we don't think that we're not trying to. What we want is a physical knee simulator that feels like a knee. When an orthopedic or any doctor is training in terms of how to use, how to do one of these tests to see whether or not a ligament is torn, it is essentially all proprioceptive feedback. So they pull on the ligament and they are just feeling, they literally will compare your healthy knee to the one that you've injured, and they will develop some sort of intuition of it feels looser than the other one. Now, the fidelity of that, right, for someone who's not a specialist is pretty low. And so that's why we think there's complications in that process, because everyone's different. We have variation in terms of the population. And so we need to standardize that. And so it's not so much about whether it's biologically functioning like a ligament, it's whether or not mechanically the reaction force you're getting is what we would expect. And so in this case, we're not really as concerned about biological function. So we're just trying to find a material that works.
See. Thank you. And
yeah, Mariana, Amy has a question that I had as well, which is how hard is it to translate some of the sheep and horse work to humans? Is there a big barrier in carrying it out? And is it more of a funding barrier?
I think what our hope now is with the sheep and even with the equine data, is that we can actually develop a predictive remodeling algorithm that might be translatable. So the question, you know, there's some issues in translating, for example, what we know about exercise in rodents, which don't have osteons, so their bone structure is quite different from larger osteonal mammalian bone. So that's the critical gap there. And so what we're hoping to do is fill some of that by doing these studies in larger animals. But then beyond that, you know, whether or not we can really develop a predictive model that would say, if we applied these set of muscle forces, we would expect this strain energy, and then we test it and see if we actually get that adaptation. And I think then we could possibly, right, if we have this algorithm that at least works in large osteonal bone from large mammals, that might be translatable to humans. The challenge in humans is, of course, doing exercise when young. It's not easy to get the imaging data that you would need to assess the properties. So radiation essentially prevents us from doing a lot of these growth type studies in young children, right? I mean, from an IRB point of view, this is almost impractical.
Thank you.
Mm-hmm. Can the ligament be detached so the clinician can compare motion with and without the ligament? Yes, so our hope is actually that we'll be able to essentially loosen the ligaments on the fly. In the worst case, you know, scenario, in our first sort of prototypes that we're looking at, we are essentially developing different ligament constructs that represent an intact ligament, a partial tear, and then a full tear being almost essentially no ligament. And so the goal, right, at the first level would be you'd have 3 simulators, and then a clinician would essentially need to test all 3 and identify which is which. The ideal version of this is that could even be done within the single simulator. And what's pretty exciting, and I didn't talk a lot about here, is the fact that we're combining this with augmented reality. And so what the clinician actually sees is nothing that's the actual physical object, the actual constructs. What they see is a representation of the actual ligaments moving, turning red as they're being stretched. And so we think that will also help in terms of the learning outcomes there.
So is it more beneficial to have straight bones or slightly curved? One question from the audience.
That is such a good question. Yeah, I don't know. I mean, there is the question of why are our bones curved, and whether or not, like in the case of the sheep, the femur is much more curved than in the case of humans. We're bipedal, and although there is bending of the femur, our bones probably see much more compressive loads than bending. But in the case of the sheep, you know, some of that curvature might be beneficial. But, you know, bending is related to the moment of inertia, so the distribution of the bone within a cross section might also, I would say, be equally important.